screen, so that's fine.
Hi, thank you so much for inviting me to this webinar. My name is Vinay. I'm working as a Chief Operating Officer at NSRCEL Sheet India Private Limited. So, okay. So this is our whole mission.
There's a background noise. कुछ कूट रहे हैं कोई कुछ, तो आप वो दरवाजा बंद कर दीजिए जहाँ से वो आवाज आ रही है, please.
Okay, okay.
हाँ जी.
Okay. So, yeah, okay. Do you ever realize what kind of problem have been facing by the woman goat farmer when he entered to the village or local hard? She has to pay the lots of taxes while entering the herd. Then she has to sell their goat with the on the basis of trader trial error method. This just kind of estimation. But while doing this work, they are always facing the lots of challenges. While entering into the market, the transportation cost will be the huge challenges for the Pashu Sakhi, and some point of time they get cheated due to no price assessment, and they get disease to the life, get disease when the goat is not sold into the market. So and how Pashu Bazaar changed the situation, few of them. So with the same mission our organization is trying to organize the unorganized market of the goat trading. So that this method, live body weight and grading weight estimation, farmer get at least 10 to 15% higher rate as compared to the traditional method of trial error by the local trader. These are the challenges which,
I think
yeah,
it's enough. She can do it in the.
Yeah, we can do it later. This is just a check. Thank you.
Just
yeah.
Use the background or do the thing with your camera so that you can come in perfectly.
Okay.
Yeah. So because there is time for your session, right, when you're going to speak. So now I'll go over to Professor Babu Subramaniam, the keynote speaker for today. He's the professor and dean from VIT College, Vellore, and their focus is on agriculture. So he's going to talk today about innovations in agriculture and especially innovations that they're doing in their institute. So Professor, this is not the actual session. We'll start the actual session in 8 minutes, but this is a trial. So I'm introducing you to the speakers. Yes, over to you, sir. This is just a check, and please try sharing your screen, Professor.
Yeah, sure. So is my slide visible in full screen? Okay. Very good morning. I am Subramanian Babu, Professor and Dean of VIT School of Agricultural Innovations and Advanced Learning, Vellore Institute of Technology in Vellore. So this is going to be sharing of some agricultural technologies, what we are working on, what we have developed, particularly with regard to the organic farming, how the science and technology can help the organic farming to go more precisely and improve the yield, because organic farmers have a problem that their yield cannot match with the modern farming of inorganic farming. So I will be talking about some of the technology. So this is the one, that is the precision organic farming. I think my slides are moving. Okay. So I'll be talking about our work and technology development in the plant growth promotion, as well as the pest and disease management on crop protection. What are the products what we are developing? I will be sharing through this presentation. Thank you.
Thank you so much, Professor. That worked perfectly. So please be on mute. Request all the speakers to be on mute, and please keep your videos on. We'd like to see you. Right, and we're just going to start the webinar in a minute.
लोग मीटिंग कराते हैं, बताते हैं ये आपका इतने का, उसको तौल के भी दिखाते हैं कि देखिए आपका ये इतने का। तो जो वो 3,000 में ले जा रहा था, वो अब सर लोग तो 5,000 दे रहे हैं, 5,500 दे रहे हैं, तो किसान भी खुश है, हम लोग भी खुश है कि चलो कम से कम हम लोग का फायदा तो हो रहा है, सर लोग की वजह से तो इतना तो फायदा हो रहा है।
अच्छा, तो आप NN ब्रीड्स जो कंपनी है जो आपको ये सहाय दे रहा है, तो ये 4 साल से आप कर रहे हैं, है ना?
जी, ये तो पशु बाजार तो 4 महीने से शुरू हुआ है। और
अच्छा।
वैसे जी 4 महीने
अच्छा।
से हो रहा है। पहले तो पशु सखी थे तो उसमें मतलब इलाज करते थे, लेकिन अब ये 4 महीने से जो शुरू हुआ है, इससे बहुत मतलब लाभ हो रहा है। और उसमें सब लोग ये भी बताते हैं कि जैसे डिवर्मिंग करा दिए, टीका लगवा दिए, तो पशु भी अच्छा रहता है, तो और दाना खिलाए तो उसका वजन भी बढ़ता है, तो उससे ज्यादा लाभ भी होता है। ये भी बहुत फायदा हो रहा है किसान का, तो किसान भी खुश हो जाता है कि चलो हमको इसको भी बता रहे हो, बढ़ भी रहा है, पैसा भी अच्छा मिल रहा है।
अच्छा, तो आपके साथ और कितने लोग ऐसे काम कर रहे हैं आपकी तरह, और कितने पशु सखी है
आपके प्रोजेक्ट में? आपके साथ कितने पशु सखी है टोटल? मैं बताता हूँ, मैं
50
आपके साथ।
आपकी तरह और बहुत सारे लोग होंगे ना?
जी जी
हाँ, तो और कितने लोग हैं?
50 लोग हैं लगभग पशु सखी सर, जो सब लोग काम कर रही हैं इस पे।
अच्छा,
अलग अलग गाँव में सब लोग कर रही है काम।
अच्छा, तो ये मंडी जैसे पशु बाजार का काम होता है, वो कितने बार होता है? महीने में एक बार होता है, हर हफ्ते होता है,
2
कैसे
बार
होता
होता है, महीने में 2 बार मैम होता है इसकी मीटिंग। और 2 बार हम किसान को बुलाते हैं, वो सब दिखाते हैं, जिसको जैसे बीच में भी जरूरत पड़ी तो हम सबको बुलाते हैं कि इनको जरूरत है, आप देख लीजिए, तोल लीजिए। तो सर आते हैं, तुरंत उसको देखते हैं, तोलते हैं, फिर जितने का धन होता है उतना पैसा देते हैं।
बहुत बढ़िया। थैंक यू जी, आज आप आए और आपका जो एक्सपीरियंस है, जो इनके साथ NN ब्रीड्स के साथ काम करने का, आपने हम सभी को बताया है। अभी अरुण जी, आपने जो बताया है वो इंग्लिश में सभी को थोड़ा सा ऐसे पूरा आपने जो अभी तक बात की है उसको मिला के वो बताएंगे, ताकि सभी लोग समझ पाए। ठीक है, शुक्रिया, आप आए आज और अपनी बात बताइए। ओवर टू यू अरुण, please summarize what she has shared.
नमस्ते।
नमस्ते।
मेरा नाम अरुण है, and I would like to summarize Ms. Kesar Jahan's experience working with Pashu Bazaar. What she had said is, in the earlier market where there was local traders by the name of Chikwa, they used to do guesswork while purchasing from the farmer. So they do not have any objective pricing mechanism. So they used to come to the farmer, tell that this goat is not looking good, or it is not of good health. I'll only give you 2,000. I'll only give you 1,000 for this goat. And sometimes they used to wait. They used to tell the farmer, "Ki I'll give you 5,000 today," but after 2-3 days the goat would get sick. That time they will go to the farmer and say, "No, no, now the goat is sick. I'll give only 2,000 to you." And the farmer is forcefully, selling the goat, because if the goat dies, he'll get 0 rupees. So 2,000 is a better choice, so he gives to the trader. Pashu Bazaar, the work we do is we weigh the goat. We have live body weight mechanism of pricing. So once we weigh the goat, we understand the weight of the goat, and along with the grading mechanism, we have an objective pricing which we tell the farmer, and the farmer can decide whether he wants to sell to us or the trader, if whichever is more. So with this, what happens is there is no guesswork. Farmer is assured that whenever we tell whatever price is coming up, that is by the weight of the goat. So after 1 month also, if the farmer comes with the same goat, the pricing will not change much. So the farmer is assured that this is the price he'll be getting. Another thing is, Kesar Jahan ji has been doing treatment, deworming of goats, vaccinations, etc. for the past 4 years. But the farmer has never been motivated to give medicine to his goats. But when Pashu Bazaar started giving good price for a healthy goat, a goat with good weight, that is when the farmer is motivated to buy vaccination, buy deworming drugs for the goat, so that it helps improve goat feed also. So because of through that, Kesar Jahan ji's business of goat feed, the vaccination, this is also increasing to the farmer, And in the long term, farmer is having healthy goat kid because the market is improving. So this is a difference that Pashu Bazaar is bringing across Kaisa Jahanji, like Pashu Sakhis and the farmers in her village. So with Kaisa Jahanji, in her we have project-wise allocations. With Kaisa Jahanji, there is 50 Pashu Sakhis, but in overall Lucknow, UP, we have more than 300, and across Bihar, across India, we have more than 2,000 Pashu Sakhis with us. Thank you.
Thank you so much, Arun, for summarizing what Kaisa ji has been sharing with us. Kaisa ji, namaskar. शुक्रिया आप आके आज बात करके आपने सभी को समझाया कि आप कैसे काम करते हैं और अभी आपको कितना फायदा हुआ है इनके साथ काम करके. So it's an information for everyone that who's looking at a better pricing, especially for somebody who's working like Kaisa Jahan, who's a Pashu Sakhi. So please reach out to NN Breeze. They will share their details and we will go over to it in a little more detail in some time, right? And thank you, Arun, for summarizing all of this. Pleasure hosting you and Kaisa Jahan also today. Namaskar. Thank you. You can be on mute and you can continue. Thank you.
Thank you.
Over to the most awaited speaker for today, Professor Babu Subramanian. While he is the professor at VIT and College of Technology, Information Technology, their focus is on agriculture. And today he is going to share with us about innovations that have happened and in the startup ecosystem, especially from their institute. So it's a pleasure to have him here again. He's been very actively involved with us on the Kisanmitr initiative. Over to you, Professor Babu.
Very good morning and thank you, Da, for the nice introduction. So, I'm Professor and Dean heading the VIT School of Agriculture and Innovation
and Advanced Learning in the Vellore Institute of Technology, Vellore. And this agriculture school is doing not only the education and training, but also innovations related to the technology required for the farmers. We are closely associated with the technology business incubator in our VIT, which is also helping us for the supporting the startup system. So I'll be talking about two important technology what we are working with. One is with regard to the soil nutrient management in organic farming. So in the organic farming, there is a concern at the field level by the farmers who adopt organic farming practices about the performance and the consistency in the yield of the crops. And it is not comparable with that of the inorganic farming where we use chemical pesticides and fertilizers, which gives immediate effect. It is because most of the organic manures and inputs what we use, they release the nutrients slowly, and therefore whatever the manures that are applied for a given crop will be only beneficial to some extent for the standing crop, and it goes on helping the succeeding crops. Therefore, the nutrient management precision is very, very important. This Liebig's law explains about the importance of individual nutrients. So in this barrel, every stave, we consider that it is a given nutrient, maybe nitrogen, phosphorus, potassium, calcium, magnesium, etc. So if there is a scarcity of one nutrient, that will lead to the yield compromise. And if we repair that particular stave and fulfill by the required nutrient by supplying a particular manure, the other nutrient which is scarce will affect the yield. So therefore, precision nutrition and the complete nutrition required for crop is important. And for this only, we are working with the sunflower, and we have developed a technology to optimize the required nutrients for sunflower. So this can be done for all the crops. For example, what we did was we analyzed the nutrients in the soil. We analyzed the nutrients that are present in the manures what we are applying. We apply the farmyard manure, neem cake, and other oil cakes, bone meal, vermicompost, etc. Then we also have the statistical data available in the government that tells us what is the ability of the sunflower plant to absorb nutrients during different growth stages. Therefore, after applying the menus, we studied the release of the nutrients over a period of 60 days. Those are the critical days for sunflower, which has a total of 90 to 100 days lifetime. So based on this, we used those data and developed some formula that will help to determine how much of the organic manure to be applied to get the required NPK for the sunflower crop. This is exactly to replicate the inorganic farming where 60-30-30 kilograms of nitrogen, phosphorus, and potassium is recommended for sunflower under the irrigated condition. And those nutrients, whether they are supplied by the organic manures, is now precisely calculated. So the organic farmers basically apply in random quantities and in random mixtures without the much knowledge about whether the nutrients required for a given crop is supplied. So for this, we have derived this package of practice where based on the target yield, for example, sunflower, usually the PSA5 variety developed by Tamil Nadu Agritech University, maximum yield reported is 1700 kilograms per hectare, whereas we can fix at a higher target of 2500 kilograms and go for a calculation of what is the combination of different manure and how much each is required to attain this particular yield. So this package of practice we will be soon taking to the farmers for the trials in their field, and we are also now concentrating on the other crops like rice, maize, and vegetables, etc. This is with regard to one technology. The second one is about the next generation bioinputs for agriculture. With our industrial collaborator Farmers Biofertilizers and Organics Limited at Coimbatore, we are developing products that are useful in agriculture. The bioinputs in agriculture include plant growth promoting bacteria, nutrient mobilizers for the plant protection, for the pest and disease control, etc. But you know, for a long period of time, more than 30 years, 40 years, we have been using the same trichoderma, Pseudomonas fluorescens, Bacillus thuringiensis, phosphate mobilizing Bacillus, etc. But the problem of using the current bioinputs is the inconsistency in the field performance. So a given biocontrol agent may not work in the all agroclimatic zones. It may not work in the all soils in same way. The functions and the efficacy may differ according to the crop and according to the microbial native microbes that are present in the soil where the bioinput is applied. So there is an inconsistency that is felt in the field level by the farmers, and therefore we have a technological solution for that, exploring the native microbes. The exploration of native microbes means that we can explore from the same field or the area, we can explore from a crop specific way, and we can also explore from a family of crop specific way. I will give one example each. So the mulberry crop that is done in our Vellore and Tiruvannamalai districts in Tamil Nadu. So they are getting affected by Fusarium wilt disease. Therefore, our team went and isolated the rhizosphere soil, and they have characterized 12 different Bacillus species. When we put them together in the form of consortia and develop a product and gave it back to the farmers applied that, and they found that the Fusarium is controlled. That means the biocontrol agent or the microbe is present in the same soil. However, the population or the number is not enough to fight against the Fusarium. Now, when we develop that into a product by mass multiplication and put it back in the soil in huge quantity, that means huge population or concentration, this product is able to fight against the fungal disease. So that is called as the native microbe for the biological control. We have started with this mulberry. Second thing is about the native microbe from the specific crop. For example, onion rhizosphere has a specific microbe, Streptomyces lidicus. When we mass multiplied and applied, it gives a very good effect in the plant growth as well as the pest and disease management. And the third example is about the endophytic microorganisms. There are thousands of microorganisms living inside the plant. So in cucumber, we have started this project and we have characterized all the bacteria and fungi that are associated inside the cucumber, and some of them are known to be beneficial in other crops as well as in cucumber as per the literature. We are now isolating and making a consortia that will be applied to cucumber, so that during the seed stage itself, when the seed germinates, these microbial consortia will occupy the growing plant, and they will establish inside the plant and fight against the unwanted pathogens, as well as they help in the growth because they are good hormone producers. These are the three products in the bio inputs we are working on. And lastly, this is not a technology, however, I would like to share the program what we have for our students. There is a program we introduced which is unique to our institute called Farmers Attachment Program. When the students finish, they come to the third year of their PSC agriculture, we attach every student with one farmer. So the farmers, when they have some field level problems like pest problem, disease problem, water, soil problem, etc., they communicate to the student. Student discusses with the faculty members or the scientists, and together we arrive at a solution and give back the solution to the farmer within 48 hours. This is the way by which the student learns in a two-way process. Student gets the experience of the farmers' field level problems also, and also gets the scientific information and technological solutions from the scientists. So this is one such example that the rat damage in paddy, how the students and faculty were involved and how they gave the solution. This is a successful program in our institutes. With this, I once again thank Mukta and all the officials of the Kisanmitr program for giving me an opportunity to share some of our technology innovations happening in Vellore Institute of Technology. Thank you.
Thank you so much, Professor Babu, for sharing details. And after having him over, he's both the dean as well as teaches, so he holds the responsibilities in terms of the management role, and also I think is backbone for many of the things that happen at VIT. Now, thank you for your time on a Saturday morning and to share details of the innovations that are happening through your institute. We'll go over to the next speaker for today, Dr. Meenakshi. Dr. Meenakshi is a former professor and head animal biotechnology. She has an amazing product and something that will change especially the allied spaces in a different way. Dr. Meenakshi, over to you. Please go ahead, ma'am. So while we wait for you to share your screen, you're on mute, ma'am. You need to unmute yourself. Yeah, we can hear you now. Please go ahead.
Thank you very much for inviting me on this platform to share my findings or my views to the audience to this platform. Good morning to everybody. My topic is, I'm just sharing my slides.
We can see you, ma'am. Please go ahead. We can see your screen. Yeah.
So I'm going to talk a little bit about the gold nanoparticle-based test for rapid detection of foot and mouth disease virus. It is based on the structural proteins which is present in when infection is there. We know that this is an economically important disease listed in the OIE list for important for import and export trades of animal products and germ plasms. And it covers a wide host range, including cattle, pig, sheep, goat, yak, water, wild animals. And it is, if we see the direct cost of loss due to this disease in animals is loss of production in case of large animals, milk production in case of sheep and goat, wool production is lost, loss of animals in case of mortality when caused by this disease. Indirect cost occurs due to eradication cost. Once disease comes into the disease-free zone, then lots of restrictions are there on the international trades. Eradication cost is there, loss of trade and exports, lots of affiliated industries, and consumer fear is there. But fortunately, this disease is not transmitted to human beings. It is animal to animal, from domestic animal to wild animals. And that's why this is very important to whenever there is, and this is endemic in India, means we have lots of pockets in India where this disease is very, very common and stay there for years together. And if we see that the economic losses incurred by this disease is 655 to 21 billion USD per year. And for the countries where this disease was not there, for example, UK, where there was in 2000, after 2004, this disease entered into the country, over 1.5 billion years per year's cost of maintenance of this disease is there. Virus is very small picovirus virus, and normal RNA virus. So we know that RNA viruses, they undergo lots of mutations. That's why we have 7 distinct serotypes, OAC, Asia1, SAD1, 2, 3. But in India right now, we have most common is the O followed by Asia1 and A. More than 60 strains are there, huge genomic diversity is there, and there is no cross protection. And I just want to add here that I was associated with the All India Network program on blue tongue, where for the first time we developed the vaccine. For the past 30 years, we didn't have any vaccine. And in 2015, we transferred the vaccine technology to Indian Immunologicals for pentavalent inactivated vaccine for blue tongue virus. Now it is giving good protection to the animals. Farmers are happy, and we know that in South India, this disease is very common, and this disease is common in northern part of India as well as southern part of India. And vaccine, though vaccine is there, but protection immunity is very short-lived. Up to 3 months, this virus is very hard. It can survive up to 3 months in the environment, and cold climate, it can survive longer. And if sunlight is not there, it still stay longer for years together. In milk and in dry milk powder, it can stay for years together. Inactivated at pH 6 to between 6 and 9. So pH is very important, and it can survive in bones, glands, milk products. So a very hardy virus. So whenever there is outbreak, so we know that buffaloes, they are very, very expensive. In lakhs cost is there. One buffalo dies for a farmer means a big loss to him. So if we have, when there is outbreak, or even before outbreak, when virus is in very small amount, we can detect it, so we can prevent the further outbreak or spread of the disease. So we have many traditional tests like PCR, real-time PCR, we have sequencing-based tests, but those tests are very, very expensive, time-consuming, very laborious, lengthy, and need special expertise. But this test is very simple. It can be even the field veterinarian or even the farmer, progressive farmer, he can learn how to do this test. So this test, that's why this test is very unique in itself. It is gold nanoparticle based. Gold, this salt is available in the market, commercially available, and very less amount is used by this. And we synthesize these gold nanoparticles in our laboratory, and then we characterize it. By TAM and SAM, you can see here that these are the small particles in nano scale, and then we have conjugated these nanoparticles with the antibodies, primary antibodies, and then conjugated particles can be used for detection of the presence or absence of the disease in the samples, biological samples. And this is how the size varies when it is conjugated with the antibody. Size is 16 nanometer, and it changes to 18 nanometer. And in the SAM also, you can see the change in the structure of the these nanoparticles, aggregation of the nanoparticles. And before conjugation, we have to purify the proteins. And this is the picture showing the purification of the protein by STS page. And you can see here the light and heavy chains of the immunoglobulins. And performing the test is very, very simple, that we have to use nylon membrane, or we can use the nitrocellulose membrane. And we just extract the RNA antigen from the known sources, and then coat it onto the nylon membrane, dry it for 15 minutes. Then we have to block the rest of the sites with the BSA, 2% BSA. Then after 15 minutes, we have to put sorry, I have put it wrong. So we have to add the sample, just 1 microliter of the field sample, then wait for 15 minutes, then wash the extra superficially attached serum to the membrane. Then we have to add just 1 microliter of the gold nanoparticle conjugated to IgG onto the membrane, wait for just 2 minutes, then wash away the extra attached, and then see the result with the naked eyes. So you can see the result with the naked eye. No specific instrument or any else is required to see the these visual changes. So this membrane shows that we have, this is the negative control, so there is no reaction. There is the normal horse serum, means heterologous serum we have added. No cross reaction is there. Then we have the normal bovine calf serum is there. No reaction is there. This is the positive control, means antibodies are present against the FMD. So we can see here the dot, and this membrane shows the 30 samples result from the field sample we collected, and you can see almost 50% samples are positive by using this test. Within you can see within half an hour or within maximum 1 hour can be used to detect the samples positive or negative. And further advancement has been carried out for this lateral flow assay. The same thing can be performed in a small you can see this small strip, and we know that everybody now knows that these type of tests are commercially available for many other things like sugar and pregnancy test. So just like that, we can, if one line is there, it means sample, this sample is working properly, test is proper. And if 2 lines are there, one line for the control, this is control, and lower line is for the test line. So if 2 lines are there, sample is positive for the FMD disease, or we dwelt initially this for the blue tongue virus, and so it was then followed we dwelt for the FMD because we have lots of problems in our state also. And this is how we can perform this test, and visually anybody can see, even layman can see plus or minus on the membrane. And that is how we do this test. And I'm thankful to our university and our administration for giving me this opportunity to do this test. And moreover, the ICR under All India Network program, under which I dwelt this test, which was the mandate also, and FMD was the extra work we did for this. So thank you very much. Thanks to all. Any question, queries are welcome. Thank you.
Thank you so much, Dr. Meenakshi, for sharing such an important detail of how you've come up with an antibody with the virus. Please share the details of your mail ID and probably your number. There might be people who might want to reach out to you for the solution, right? If it's on a web page or something, then please share it in the chat so that
I have shared my email in my
slide. But after this, then there are other speakers also who will be speaking, right? So if you share it on the chat, they can reach out to you.
Sure.
Thank you so much. Professor Babu, thank you so much for your time and for sharing this today, Dr. Meenakshi. Professor Babu, may I request you to share details of your coordinates as well, so that the people who want to reach out to you will do the same?
Sure.
Yeah, thank you so much.
Thank you. And thanks to all.
Thank you. And now over to the next speaker for today. After Professor Babu's keynote speaker address and Dr. Meenakshi's product on antivirus with such a specific niche solution, we will go over to Mr. Vinay ji. Vinay is the Chief Operating Officer of NN Breeds and Seeds Private Limited. And he's going to speak about transparent and equitable trading of livestock. And I would like you all to be, a little more aware of what's happening in this space and how you can reach out to them as well. Over to you, Vinay ji. Please go ahead.
Thank you so much, Mukta. First, I would like to thank you for inviting me to the Kisanmitr program. I'm Vinay. I'm working as a Chief Operating Officer as NN Breed Seed India Private Limited. But before my starting career, I was work as a trader, just like a chikwa in one mandi to another mandi. And then I come with the contact with the Go Trust, and Go Trust as a social change fellow. Then I shifted to the company, this Nandi Nandan Breeds. So in Nandi Nandan Breeds, we have a business of live goat, and with the name of PashuBazaar.com. So this is our vision, which we wanted to change the unorganized market into the organized market. So do you ever realize what kind of problem have been facing by the small goat farmer, especially women in Mandi? Even you can guess how she has to pay the tax at the heart, transport livestock, and get cheated due to no pricing assessment, and get disease to livestock if he return home without selling a single goat. And how Pashu Bazaar changed the situation for the few of them. So with the same mission, which I already said, organizing the unorganized market of a goat, we have started the live body weight and grading based estimation at a farmer level. So the farmer can get 10 to 15% higher rate as compared to traditional method of a trial and error method by local trader. These are the problems in the physical heart and the solution provided by Pashu Bazaar. When I see the point number 1 and 2, the irony of sale and purchase of a livestock like a goat is we can purchase today anything like car, agriculture products, sheets within 2 or 3 hours, but not in case of goat, which will take at least 3 to 5 days even for a single lactating goat. Problem lies in two fronts. One is standardization of pricing based on objective criteria and unavailability of information of seller and buyer in a geography at the particular given time. Rate are subjected to the estimation and palpation of a local trader through trial error method, but not known to the producer who invested over the year to produce his product. In our live body weight and grading based estimation is help to provide this get an surplus sellable goat and right amount of price to the farmer level. In point number 3, we can see in physical heart there is no quality parameters and the genetic information about the livestock, but in our model we have objective grading based assessment such as length, girth, circumference of the goat, and at least one generation reproductive history, so that producer get good price of a productive goods. In physical heart there is no traceability in terms of fund and total cash based business.
You might want to scroll down your pitch as well. We're still on the first sheet.
Actually, this is for first sheet. We are talking about.
Okay. Yeah. Please go on to the next so that others can see it and share it in the PPT mode. No, you might want to increase the size.
Okay.
Please go ahead. Please continue.
Thank you. So in physical heart, there is no traceability in terms of payment billing system, and Pashu Bazaar providing the platform for offline, online payment and purchase billing system. In physical heart, you can see there is a high probability for a viral disease because all huge number of livestock kept into the one place. And in our Pashu Bazaar platform, virtual market linkage at a one place where buyer or purchaser come to a virtual platform, and there is no need to kept all livestock into the one place. And there is a zero women women participation. But in our model, we have a Pasu Sakhi, we have a goat farmer, and direct sell purchase with support of this model. So when we said the live body weight pricing estimation the first question is arise. How to
Vinayji, one
Yeah.
small request. While
Yeah.
you continue, please start sharing your slides, because in few minutes, for the time constraint we'll have, just ensure
Okay.
that you get on to the topic
Yeah.
as well. Yeah.
Is my slide is
Yes.
visible?
Yes, it is. Please
Okay.
go ahead.
Okay. When we see the first point, so we have to understand the difference between the empty stomach and filled in stomach of a goat, because we have to estimate the actual price of a goat, not the feed inside the stomach. That is why we have to follow the first process. In second process, we have to understand the present meat estimation by three trader of a same goat and find the consistency of their estimation, because these are a trial error method. So we have to check.
Vinod, please share your slides. No, we are not able to see. In your stream, no, we are not seeing. No, this is your display. Is not.
Hey, wait a minute. Wait a minute.
Vinay, please continue. Atif will share the screen on your behalf, because
Yeah, I guess there is some.
yeah, no problem. Atif, can you please share Vinay's screen? Vinay, please continue speaking what you were sharing and continue, because we are short of time. Please continue with what you're sharing. Yeah, go ahead.
Okay. So first is to assess the difference between empty stomach and filled stomach of a live body weight of a goat, because we wanted to estimate the rate of goat only, not the feed which is inside the stomach. And after the empty stomach weight, we want to understand the present meat estimation by the three trader of a same goat. Atif is a strategy part. Is the fourth slide. Yeah,
Okay.
Thank you.
Awesome. Thank you so much, Atif.
After the meat assessment, we have to track the real sale. We can see the with the third point. After the meat assessment, we have to track the real sale data of a traded goat in the same cluster. In this process, we have to note down amount received to the farmer and weight of a goat at the time of sale. In the next process, we started profiling the various nearby herds, understand the difference between farm gate price and hard price differentiation on the live body weight basis. And lastly, we have to understand the meat ratio by slaughtering of 100 live goat with a different age, sex, and breed. Next slide, please. So these are our working model. When we provided the rate chart to Pasu Sakhi, so Pasu Sakhi start doing the rate declaration on daily basis. They go to the village, declare the price of the goat for the goat farmer. If any goat farmer wanted to sell their surplus goat, they inform to the CLM. This is the next level in a form of code. You can see this code is M2A16-4600 something. M2 means this is a male, 2 teeth, A grade, 16 kg, and 4600 is the as per the live body weight rate.
Vinay, you will have to come to the final in a minute, because we have to go over to the next speaker.
Okay, this is our fine. Yeah.
Okay. This is really wonderful.
Okay. Then after the when CLM qualify, inform the Pashu Sakhi, yes, you can book. We have the surplus order. Then these goat shifted into the village level trade center. Village level trade center having the capacity of 10 to 20 goat at a particular time are after when we have a surplus goat around 50 to 100 goat at a village with different village level trade center, then is shifted to cluster level trade center. This is a capacity of this is they having a capacity of 50 to 100 goat at a time. During this process, procurement manager providing the bill to the Pashu Bazaar, which is a central marketing team, and central marketing teams provide the all fund as per actual live body weight in account of the cluster level trade center. So the cluster level is managed by the CLM or goat entrepreneur, and the village level trade center will be managing by the Pasu Sakhi or husband of Pasu Sakhi. These are the whole model. With this model, we have sell around 20,000 goat to 20,000 goat till the date.
Okay.
Yeah.
Thank you so much, Vinay. We'll just go to the last slide, please.
Yeah, this is our team, which I already discussed.
Okay. Atif, over to you. Thanks, Vinay. Atif, you're on mute. We can't hear you.
Yeah. So I want to thank Mr. Vinay for the presentation. I want to welcome Mr. Dr. Raghavinder Singh. He's a principal scientist and head division of animal physiology and biochemistry, ICR Central Sheep and Wool Research Institute. So over to you, sir. So should I share the screen on your behalf?
Yeah, please. Yeah.
Please start talking about your innovation, sir.
Yes.
Thank you very much, Muktaji. On behalf of our CSWRI, we would like to first thanks to our organizers, Muktaji and team, and Kisanmitr, and also welcome to all participants. And we'll take 10 minutes to just explain this one of the technology of our institutes, which is estrus synchronization and artificial insemination in sheep. Why this technology actually required now? This is actually very big in two words, I should say. This is the commercial sheep farming, which is inevitable in future, which is required for people who are coming for sheep farming system in a commercial manner. So this technology actually what we did it. This we have gone for trademark. We already taken with this technology also under the patent by our institutes, and this technology having, please go back there. This technology, this is our Avika seal name is there. So I like to just highlight it. This is available in the name of Avika seal, which is given a trademark, and that includes this is sponges, which is actually required for the question of our interest also. So this is a sponges, and this is a plunger, and this is speculum. These three things actually essentially required, and these sponges are medicated with the progesterone hormone. So this kit actually available in the form of a 24 sponges, and with these two accessories, and then in the one this kit is having a 628 rupees, having 24 sponges. So 24 animal can be synchronized by using a one Avika seal. So this is the next, please. See, this is a very simple technology. What actually we do it here, what standardization was done, that these sponges which was impregnated by the progesterone in the sponges, we put in the sheep in a first day. All the sheep, whatever sheep available, we want to make it artificial insemination in the sheep. So we want to synchronize for 1 day. So that is actually the day all the sheep bring in the one place, and then make the sponges, insert the sponges in through the vaginal tract, and then afterwards first day to start up to the continuous 12 days, and then after 12 days these sponges were sponged out from the animal, and then the same day if they are non-breeding season, then we can put that 200 IU PMSG. This is one pregnant male serum gonadotropin hormone, and then after within 2 days, 36 hours and 48 hours, we used to do the artificial insemination, and then the animal sheep get rid of from all this exercise, and then you can see the afterwards pregnant or not pregnant, that can be skinned after next 2 week after, because the cycle of the sheep is 15 days. I think this actually it's okay. It's like it's a 14 or 15 days. It's coming fine. So this is actually the one protocol which actually being a principle for these things. And after what is happened here actually, why just to like to just explain in 1-2 words. The progesterone is actually doing in the uterus mimic the progesterone in the uterus. So what happened? That actually what happened? All the progesterone continue up to this hour. So all the animal come to the day when you out the sponges, then they get. What is the advantage of this thing? Synchronize the estrus in recipient Eves, those animal, those sheep which you want to synchronize it, and perform large scale of AI. So afterwards, fixed time AI means after 12 days, then within next 48 hours we can fix AI. There is no need to see it is a heat or not. That you can directly you can after this protocol we can do the AI artificial insemination within a 48 hours by the chilled semen. And then what importance or what I'm telling, the reducing the interlambing period. In 2 years we can take a 3 crop, which is not possible in general. So in the farming of commercial farming, we can go for the having a one more crop, additional crop from the seed. So this is and allow this Eve to be grouped into the desired lambing. Suppose you want to do the breeding of your sheep in the particular season, particular time, so the lambing can come on a particular festival. So such type of you can design the breeding of the sheep by your own way. And most important thing, you can save the labor, easy management, and harvest the at a time all the lamb in a very small period of time. Within a 2-3 days, all the lamb get lambing and happens, and then you can take it out in a all management practices in together. So this type of this many others this advantage we have, but like unstressed animal which remain out for the farmer, they can also bring in the pool for the breeding. So unhealthy animal, they are not good, they can be treated for 1 week, and then they can again bring to the in the breeding system. So this technology have a many advantage, and really this is a time to come. We have to go for this technology. Now we are still very much demand for this technology in the sheep, because we know that 7.5 crore population or sheep number in our country, and that is also demanding towards to meet out the this meat requirements and the wool carpeting system. Our country, we are leader. So this actually time to come in the southern our states now leading in the this demand for the sheep meat and other requirements. So this technology have a very commercial beneficial. See, the training we used to give within the last 2 or 4 years, we have conducted training in 2 trainings, and likewise we used to train. So 10 days course we have for the participant. They can be field veterinarians, they are livestock assistant, or they can be a supervisor also, and there are many other faculty who are really involved in a professional to enhance the sheep reproductive efficiency in the field condition, or people who are rearing the sheep, progressive farmer, they can join. We more than 250 persons we have trained in the other Uttarakhand, JNK, Himachal Pradesh, and like other southern country, southern state also. Likewise, we are also having a assessment with our Australia and some other part of other country also looking for such type of a production in a large quantity, this avica production. So next, please. So this is our we are sale the expense just for the popularization of this technology. So we are selling this sponges on that cost, which is very nominal cost. No profit, no loss cost we used to sell. And this see the that fertility rate, how it is more 80 to 90%, and the estrus response more or less 100% sheep comes to the in estrus condition. So this is also the cost of per sponge is approximate now presently is 24 rupees, and this PMSG actually required whenever the animal are in non-breeding season. Means non-breeding means then the breeding season means there's some in the spring season, and then on top afterwards if you need that you have to give 200 international unit of PMSG, one hormone which is 200 IU. We have to give it in the day of the sponge out. So this is actually how input be required, and addition to the sponges by the farmers. Next, please. See, this is after the sponge out, then we could go for the artificial insemination. Why this is important? because there is no need to see the animal in the heat or not. The day we out the sponge from the animal and then plan the artificial insemination, and they already we do this in lab. Already we have our or anywhere we can do it. We collect the semen and donor, or you can go for the natural mating also. But we're doing here artificial insemination. We can go for natural mating also, but that is not maybe feasible, no, not maybe economical. So and then prepare the AB, and this is all up to do in the our lab only. And then this is a semen dilution, and then preservation, and then semen development. This part actually goes to the in the field. So we go in the morning or evening in the concerned field area, and then we can deposit the semen in the uterus side, and then this part is completed for this course of this technology. Next, please. You see, this is the actually we did. Why is the sheep have a little problem? Because of this, the transvaginal tract of the sheep compared to the goat or buffalo or even this cattle, this is little bit tedious. This have a septum sites. So there are 5-6 septums are there, and that's why we putting the artificial insemination is little more tedious, difficult. That's why sheep in the frozen condition, artificial insemination is not possible till today. Which is not in our country, but most of our whole the world is working on this area. Still we have somewhere lacking, but liquid semen, that is a very good, we are already doing since so many years, and this is successful, and this is more than 40, 50 to 80% of the lambing is there. Why happened? So that's like I tried to show you the vaginal tract of the sheep. That is actually the problem, the diseases type of the tract. So septums is not possible to inject or to reach the uterus side. So this is a laparoscopy we did it. This is a frozen semen is already functional, but when go for the cervical mode, then it is not much functional. So this is actually problem which we are facing, but we are working on this area also. So this is the status of the our what the mode we are doing in the field condition. Next, please. Why actually like to show you this slide? These are the actually the institutes like state, many ICR institute also, state government, our universities, division, KVK, organized farm and NGOs. All they have more than 10 years continuously, and more than, if you say the overall 15 years, we have a many data. We have a large data, scientific data, that after what is the feedback of this technology in our in the field condition. So that is actually we assess that is a functional in the liquid chilled semen artificial insemination, followed by the followed this estrus synchronization, and followed by the artificial insemination. So in nutshell, why I like to explain this thing, that this technology have a not only in sheep, this can be done also in goat, it can be in the cattle and buffalo also, but we standardized for sheep in the first time in our country by this institute, by division, and that is actually very much available and implemented, and this is functional in our division. Anybody needs details, any type of needs demand and purchase, and any, we have a CD also for 50 minutes we can show, and you can see that each step how we can do it in the field condition. Even this the simple person, if you have a training of 2 days, he can do the estrus synchronization. It is not any much more difficult process in the field condition. AI is need little more you can say some like semen collection, and then we have to evaluate, and then dilution, and do it in the lab. So this is also very small things that can be done within a 2-3 days training program. Anybody can learn and do this technology. This is actually bring in the moon in the farming of the sheep farming system in a commercial manner, because we know that traditional farming of sheep is not going to be there in time to come. Within a I think within 2-3 years, 4 years, you could say significantly is going to drastically reduced because of the synchronization of the rangeland. Don't have a, there are many other problems came now. People who are rearing the sheep, they don't have that much remuneration because of marketing, other things. So now we have this commercial farming, this type of technology, not this technology. This is along with we have a more 5-6 technologies, like by how we can utilize the urine by using the abikhad. We can utilize the wool also, addition to the carpet wool. We can use for the fiber. We can have many type of boards be prepared. We have many type of the artisans we have prepared. So likewise, all the parts, likewise the milk products we are already prepared. Many milk products also, meat products also. This is also some technology we produce, which have in our institutes. So overall, collectively, the sheep animal as a whole can be utilized and can be the value of the seed can be added in the market by the farmers, so they can ultimately get more benefited and they can have a more remuneration from the seed farming kind to come in future. So this is our details. We can any have mail used to come, and then anybody can also by division. We have a mail also in our site. This is the site. We can go for our website of NRC CSWRI.
Could you please, Dr. Raghavendra ji, can you please share the details on the chat as well if anyone wants to reach out to you? Your coordinates.
Yeah, we'll do it. Thank you very much for, I think, just giving outlook to this technology. Thank you very much to you also to giving me opportunity to share with you and testing. Thank you very much.
Thank you so much, Dr. Raghavendra ji, and Professor Subramaniam. Babu Subramaniam was with us for quite some time. And Dr. Meenakshi, for the wonderful presentation, and Mr. Vinay, and Arun for chipping in and supporting Vinay and NN Breeds. A pleasure to host all of you. And Atif, thank you for chipping in and being a part of this complete webinar with us. So we hope to continue to do this. And by the way, all of this will be available in vernacular across India. So there will be more KVKs and more ICR institutes and startups also joining in to see this. So pleasure hosting you. Thank you for sharing your knowledge and best practices today.
Namaste, Meenakshi madam ji, kaise hain aap? Sir kaise hain madam? Sir kaise hain Meenakshi ji? Madam?